goal-driven attention, where low-level sort of visually salient features of the environment might automatically capture your attention, so that, for example, if there's a sudden movement, a flash of light, you might saccade, you might move your eyes to that in order to process it. Conversely, people talk about so-called top-down or goal-driven attention. The idea here is if you're finding your friend, you might be kind of scanning the faces of the people in the crowd with that goal in mind, trying to find your friend. Now, at the same time, in this situation, you're constantly using your memory, right? So, for example, if you need to meet your friend at a particular track at a particular time, you need to consult the arrivals board here and remember or encode that information so you know where to go. At the same time, you might need to remember or retrieve information from the last time you're at the train station in order to navigate appropriately, okay? Now, particularly in psychology, these two domains are studied as kind of separate faculties of the mind. That is, they're studied in isolation. But at the same time, we know just from intuition and our everyday lives that oftentimes they interact. So for example, you might be paying attention to one face in particular because it kind of looks like your friend, but you're not entirely sure, so you need to dedicate more resources to processing that particular face to kind of help guide your memory search. Conversely, a new pizza place might pop out and capture your attention, not because it's visually salient, but just because it's new. Let's say it just opened up. And so that's a manner in which, even though there's no sort of basic attentional phenomenon there, memory can kind of serve as a powerful guiding force. Now, these are kind of silly examples, but they're meant to underscore deeper questions about the interactions between memory and attention. That is, how is the manner in which we search for memories from the past, similar to how we might scan a visual environment? At the same time, how might our attention be captured not by just sort of visual features of the environment, but the degree to which they relate to things in the past? And so my talk today is going to focus on the interaction between these two systems. And in particular, in the first half of the talk, I'll be discussing the manner in which attention might operate over memory. That is the same attention system we use for scanning the environment, similar to how we search for memories or select from one of many competing memories once we retrieve them. Now, in order to first address this sort of interaction at a broad scale, I was interested in knowing just what the literature had to say about the relationship between these two domains. And so I conducted a meta-analysis just to survey the field, because oftentimes attention studies and memory studies are done in isolation. Of course, I'm interested in the interaction. And so the manner in which I did that was assessing a large group of fMRI studies. And the idea here is fMRI is actually a very powerful tool for assessing the similarity or differences between things, because, for example, the degree to which there are overlapping regions of the brain that are activated by certain tasks, it sheds insight into the degree to which they have shared or distinct computations, okay? And so I just need to orient, this is the left hemisphere of the brain, we're kind of looking at it from back and to the left. For the first portion of this talk, I'm going to focus on this one chunk of cortex right here, which is the posterior parietal cortex. I might sometimes say PPC. The other thing that I want to orient you is, unfortunately, another sort of jargony acronym. The first is this intraparietal sulcus, or IPS. This is just an anatomical landmark, just to get your bearings. And the patch of cortex more medial, so kind of the right on this diagram, is the SPL, or superior parietal lobe, or lobule. And I think that's going to be all the acronyms for a bit, so hopefully you can just hang on to those. But the general idea, those are good landmarks for some of the effects that I'll be discussing about in the next little bit. Now, why am I interested in this one patch of cortex in particular? Well, it turns out that repeatedly in the memory retrieval literature, one of the most consistent and robust neuroimaging effects is elevated activity in this region of cortex as a function of so-called memory strength. The basic idea is, for example, if you encounter something in the world that you know you've seen before, you have higher activity in this region than if you encounter something you know to be new or novel. Okay? And some memory theorists have posited that given this region's role in other domains, such as attention, this effect might be capturing so-called attention to memory. That is, when you remember something that's old, you might need to engage attention to, say, guide that memory search or select something for a specific decision. And so this region of cortex in particular is where I'll be doing the meta-analysis. The other thing I'm going to do is kind of inflate the brain. You can imagine I took a bike pump and just kind of filled this with air, and so now these darker regions are the sulci, so those are kind of the various nooks and crannies I was just showing you in that last slide. And the lighter gray regions here are the surface, the ones near the outside in that last slide. Okay. First part of the meta-analysis was to get an assessment of where in this region of the brain the so-called memory strength effects. That is, I looked at all these episodic memory studies and I tried to figure out what regions of the brain are consistently involved. And I'm happy to talk about sort of the stats and the technique I used, but kind of at a high level, the idea is I'm going to show you kind of a blob, and that blob is representative of consistent statistical effects in the literature. What we found is basically this region highlighted in blue here. These regions were consistently implicated in kind of memory strength effects, so kind of more elevated activity is a function of how familiar or how strong a memory signal was. And then you can see it's just right here and along the IPS. And then I could also go look into the attention literature and look for, say, for example, if you're cued that some target, some visual target will appear on the right side of space, you have to maintain central fixation, you kind of use your attention to attend there, and something might appear. It's that sort of study. Interestingly, what we found is these effects fall more medial in the SPL, and there wasn't much evidence for overlap. Now, in light of all this buildup, that might be somewhat surprising. If that was all there was to the story, this would be a fairly short talk. However, one thing that occurred to us is, first there was some, this is a summary statistic, so there were some studies that kind of were memory studies, but fell in these attention regions and vice versa. And the second is, when you remember something, it's kind of a multi-component sort of cognitive mechanism. And so the idea is, even though this memory strength effect doesn't seem to engage regions that are involved in top-down attention, some other aspect of memory might be implicated. And in particular, based on some of the studies we saw that overlapped, as well as thinking about this in terms of, intuitively, it's possible that you don't necessarily need attention as a function of something being old or new, but rather you need it when you can only remember a little bit about the thing from the past. So the example I gave you before is you see someone in the crowd and you're really attending them because you're not sure if it's your friend or not, and so you kind of, the idea is attention might be engaged during these fuzzy memories. Okay, so we conducted an fMRI study where we tried to tease apart different amounts of memory, that is, get people to remember different amounts of things, and see if we can see evidence for attention regions or other signs that attention might be engaged during these kind of fuzzy memories. So first, to just give you a demo of what this task was, we had healthy adults come into the lab, and they saw a list of adjectives. And for each adjective, they were cued or instructed to come up with an exemplar of that adjective. So, for example, in this demo, just to give you the benefit of kind of a practice session, I'll cue you, and I want to cue you with person male. And the idea is I want you to all generate a male celebrity that's described by the adjective. So we'll say person male, and we'll say an adjective, and then I want you to think of someone who's described by that adjective, and then just say the first thing that kind of comes to your mind. Okay, is everyone ready? Everyone's on board? I see some nods, I'll take those. Awesome. Johnny Depp. All right. So we have one Johnny Depp. It's actually been a Trump effect as of late, but historically there's Carrot Top in there as well as Tom Cruise, but that's the first Depp I've had, I think. Great, so the general idea is people see a list of a bunch of different adjectives, and they're cued to come up with either a male or female celebrity, or an indoor or outdoor scene. They have to generate something for each adjective. Now, this is just kind of the study phase outside the scanner. Critically, we move them inside the scanner, and we show them old or new words, and we have them try to remember what they had associated with it in that first round. So for example, they might see bizarre, and then they are asked to make one of 8 different responses. They could say new, saying, oh, I don't remember seeing this word at all. And that's going to give us, I'm not sure how familiar you guys are with this site jargon. The idea is correct rejections is when you say new to something that is in fact new, and misses are when you say new to something that's old. Okay? We're just going to treat those the same for here. You can say old, meaning, oh, I know I saw bizarre before, but I don't remember anything else about it. I'm going to refer that to as item only, so you remember bizarre, the item, but nothing else. Okay? You could say person or scene. The idea here is you have an intermediate amount of information. Like, I know how to come up with a celebrity, but I really don't know if it was male or female. Okay? I'm going to refer to these as kind of task-only memories. And then lastly, you could indicate if it is female, male, indoor, outdoor. And presumably here is like when you see bizarre, and you're like, oh yeah, Johnny Depp. And you could give this specific response. So refer to this condition as the specific details condition. The idea is this kind of provides bend ways of measuring different amounts of memory detail. Everyone on board? Great. And so how might we look for signs of attention being deployed as a function of these different conditions? Well, one way to do it, and it's often used in the literature, is to look at reaction times or response times. And the logic is insofar as there's higher reaction times, it's emblematic of sort of a more difficult or more effortful memory decision, and presumably might employ more sort of top-down attention in order to arrive at a solution. And what we see looking at the reaction times is this really nice inverted U shape that is, reaction times aren't the longest, for example, when you remember the most, but rather they're the longest when you kind of have this fuzzy amount of memory. That is, you have some idea that is a celebrity, but you don't quite get the full Johnny Depp. All right? So great. We think that this condition, where does this work? It does. So this condition here looks to be a nice condition that might isolate effects of attention during remembering. And what we can do is look for all the regions of the brain that have more elevated activity during this condition than for the condition where you actually remember more. And the idea is this can't be due to memory strength because it's the opposite of the memory strength direction. But it might be due to attention being deployed during memory. And what we find is nice and selectively, this region in SPL and a little bit in frontal cortex. And this looks a lot like the region we saw in that meta-analysis. You don't have to take my word for it. We did the overlap, and indeed it is the exact same regions that crop up. So that's good. We're seeing regions that are typically found in attention studies, but they're actually being brought to bear during remembering. And what's more is activity in this region tracks reaction time, not just in that crude condition-wise manner, but actually if you break it apart trial by trial, this is just a single subject, you see that there's more activity tracks reaction time across trials, and that holds even if you kind of break it down into each condition. So it's nothing, so from this it's actually, you can tell that it's nothing about, say, that relationship to fuzzy memories, but it seems to be tracking something on a trial-by-trial basis. So it could be related to something more about perhaps the uncertainty of the memory decision at retrieval rather than the amount to remember per se. So in order to follow that up, we ran a second study, also an fMRI study, and the goal here was to look at how this effect varied in terms of, instead of objectively measuring how much you remembered, just looked at a subjective measure of how confident you were about whether something was old or new. Okay, so the coding task, the study task, was still the same as before, bizarre Johnny Depp, but critically at retrieval, instead of kind of having them make this convoluted 8-point response, we just say on a scale of 1 to 5, how certain are you that you've encountered this item before, okay? And like before, we can look for reaction time differences as a function of confidence, with 5, I believe, being those kind of certain old responses and 1 being certain new. And again, we see this really nice inverted U-shaped effect. So here, this is certain new, this is certain old, so this is like, I don't know. And so this is the highest RTs. And so again, it's really suggestive that it's something having to do about sort of more difficult or uncertain memory decisions rather than the amount of things you remember per se. And what's nice is if we look in this SPL region again, we find this inverted U-shaped pattern, again matching reaction times. And I'm not showing it here, but I also track them on a trial-by-trial level as well. All right, great. So across 2 studies, we found evidence that attention might not track memory strength per se, but it still might be employed in a systematic manner while remembering, okay? But this leaves open a question, what do you mean by attention? So I'm just kind of invoking it generally. When a difficult decision needs to be made, you need more attention or less attention, so forth. But if you think about it, there's kind of different forms of attention. That is, attention varies in terms of, for example, what you're attending to. In the context of that example I gave you earlier, I've given this example, you see some person in the crowd, and you're really focusing on them, trying to sort of guide this memory search. And I'm just going to call this type of attention external attention. The idea is you're trying to get information from the environment to help guide memory search. Now, this can be contrasted with a different form of attention, and say, you know, when you're thinking about your friend, you're brought to mind the last time you met at, say, Dunkin' Donuts in the train station, and because you've done it multiple times, and this is Boston, there are multiple Dunkin' Donuts, and so you have to bring to mind, you know, the different Dunkin' Donuts in the train station and kind of select which one you were at last time. And the idea here is that this is referred to as internal attention. Okay, the idea is attention is kind of selecting on the contents that have been internally generated. Okay. So now we can turn to those studies that I just showed you and look for evidence for one form or the other. And so in particular, we wanted to test the hypothesis that these regions, just based on what we showed in the literature, might be involved in external attention. And the logic here is if these regions that we see being engaged in this inverted U-shaped pattern during remembering reflect external attention, those regions should express some other function that's suggestive of them processing external space. Now, it turns out that some regions of parietal cortex actually contain maps of external perceptual space. And I'll unpack what that is in just a second. But the basic idea here is we wanted to bring a subset of those people who did that first memory study, the Johnny Depp study we'll call it, and they came back in and we wanted to map out these attention maps and see if these are the same regions that index both sort of represent external aspects of the environment, as well as showed elevated activity during insert decisions. So just briefly, just because I don't know how familiar folks are in this crowd with topographic maps in the brain, the general idea is that there are regions in cortex where adjacent patches of cortex track stimulation to adjacent areas of external space. Okay? So the way this just works is you get your standard undergrad to come in while you're scanning them, they maintain central fixation, and you have them do this demanding attention task. Okay? So you show this kind of flickering checkerboard stimulus, and there's sort of reasons why we chose this stimulus, but the idea is you have the stimulus and it traverses the screen in a systematic manner. And it turns out that there are regions of the brain where the activity kind of tracks where in space there's stimulation. Okay? So a different way of rendering this is I'm now going to kind of color those different areas of stimulated space. And then this is not actual data, but this is just kind of a schematic representation of what we're looking for in the brain in terms of patterns of activity as a function of that external stimulation. And so, these so-called maps of visual spatial attention have been shown in the literature before, and the idea is, if you look, this is still the left hemisphere. This is our friend, the IPS, the intraparietal sulcus. So anterior front, posterior back, left, right. If we do that sort of clown motif painting here, we see this pattern of activity. And it turns out that there are, in fact, a series of maps that kind of are tiled along the intraparietal sulcus. And again, we did not discover this, but we've replicated this, has been shown in the literature. The general idea is that the visual system has many maps of external space. They're organized in a hierarchical manner, so you kind of have different amounts of processing of spatial information as you move more anteriorly in the brain. That's not as relevant for this. The chief point here is, great, we have these regions in hand, we can look to see what they're doing during remembering, okay? So I'm just going to outline them here, and what we did is interrogate each of these regions for the profile of activity during the Johnny Depp test. And what we found is a couple of them, I'm just highlighting one here, called SPL1, show exactly what we saw when not looking at the visual ROIs, but they show this inverted U shape, right? So even though this ROI shows tracking of external information, it also shows this sort of pattern of memory uncertainty during the remembering task. And so this is consistent with the notion that these regions might be doing something with external attention, that is, trying to get more information either from the environment or somehow bridging what you're seeing in the world to guiding your memory search. Just going to actually skip this next little bit. So pulling back from the first part, I've shown you some evidence that attention might play a role during remembering, and specifically the perceptual attention sort of apparatus might be recruited in order to help inform sort of uncertain or difficult memory decisions. All right, great. So turning back to the main sort of attention-memory dichotomy here, I'd now like to turn to the other side of the coin. That is, I've shown you how attention might be deployed over memory, but how might remembering or past experience influence what you process in the world, okay? And as before, I initially performed a survey of the literature just to see what was out there. Just for the sake of time, I won't really go into it, but suffice it to say there's actually a lot of effects out there that just haven't been sort of cataloged systematically. They kind of fall in the attention and memory domain. But the idea is that, sorry for the jargon, regularities, so things that are associated with each other in time, tend to capture attention. So your attention is biased towards structured information in the environment. What's more is memory for specific events or episodes will influence where you look in your current environment. So, you know, if you see the same box of cereal that you had this morning for breakfast, you know, you might preferentially guide your attention there. We also know that semantic memory is actually a fairly powerful influence on your attention. So if I ask you to hold in mind a motorcycle, and you're actually more biased to kind of look at, say, a motorcycle helmet or something like that that's semantically related in, for example, a visual search task. And then lastly, so-called working memory. You guys maybe have heard of this before. The idea is that when you temporarily hold information in mind over a short delay, it's thought to be held in so-called working memory. And people have argued that your attention is guided towards things in the environment that match the contents of working memory. Again, these are just kind of flavors of memory-guided attention. They're actually each somewhat different from one another. But today I actually want to focus on the flip side of that. So in the example I gave you earlier, your attention wasn't captured by something that you'd seen before. It was captured by this new pizza place, right? And so the idea here is the flip side of memory-guided attention, which is what promotes your attention to new things, what might guide learning of new information. And so one theory that we're really interested in testing is the fact that this is new could be defined by the fact that it's next to something that's really old, right? It's the contrast between old and new that might bias your processing towards the new thing. And so just by dint of this kind of having been old, this might seem more relevant by comparison. You might process that. And so that's the basic phenomenon that we wanted to test, and specifically we're interested in understanding what neural mechanism might be giving rise to this sort of hypothesized effect. So just to kind of spell that out and translate it into a neural prediction, you have some stimulus in the world, and say the first time you see this Dunkin' Donuts, you get this really nice high neural response. Bold is, just because I don't know how familiar you guys with these terms, bold is just kind of the neural measure that we use when we use fMRI. And sort of typified evoked response for a bold signal is something like this. So when you first saw this Dunkin' Donut, you have this really nice evoked response in your brain for the regions that care about Dunkin' Donuts, okay? Now, what happens is, next time you see it, you still get the Dunkin' Donuts regions responding, oh yeah, Dunkin' Donuts, but it's actually less, okay? And this is a very robust effect across species. This is kind of a basic property of areas of the brain in terms of if you see something that you care about, you fire a lot the first time and less. I'm going to refer to this as repetition attenuation. There's a lot of different jargony words for it, but that's my favorite jargon. But the idea is you now have this reduced or attenuated response for the third time you've seen this Dunkin' Donuts. And when this pizza place opens up next door, its evoked neural response is much higher just by the fact that it hasn't been attenuated, okay? And so the logic is, insofar as these two things are competing for processing, the unattenuated signal should receive more processing resources because of the attenuated response to the old thing. All right? So this is the hypothesis we wanted to test. And the way in which we operationalize this is we had, again, healthy adults, aka undergrads, come into the scanner, and we had them basically look at a bunch of images, sometimes one at a time, sometimes two at a time, and we had them push a button whenever any image was upside down. It was just inverted, okay? So again, I'm gonna have you all do this. If you see any image that's upside down, just clap. All right? There we go. All right, high latency, but good deep prime. So everyone saw is a fairly straightforward task. And these were relatively infrequent occurrences. And truth be told, the only reason we have those inverted trials is to keep the participant awake. We actually don't analyze those. As psychologists, we actually care about everything else that they're doing. And so, for example, you might have noticed that that last set of images I showed you had this structure. It had image A, image A, image A next to image B. Okay? This is the critical condition we care about because we care about this competitive situation where you have an old thing and a new thing. We refer to this condition as the repeat condition. The control condition I'm going to refer to as the novel condition. The idea here is you have image repeated twice, just for sort of similarity to this experimental condition, but then you have two novel images in this third competitive case. The idea here is there's not that asymmetry in old-new to kind of bias processing, so it's a basic condition. Now, we're interested about the degree to which they process this new information in this competitive situation. The way we measure that is what we call subsequent memory. And the idea is once we get outside of the scanner, you say, surprise, I'm giving you a memory test for all those items you didn't think were important. And people love that, of course. And so the idea is we show them some images they had seen before, as well as some new lures they hadn't encountered in the experiment before. And we can test their memory for the things they had seen before and look to see if their subsequent memory, if their encoding of this new information was better when it was alongside something they had seen before. Okay? Just to spell out the logic, in terms of our predictions, is this attenuated response will lead to an asymmetry so that the new information, when I then go ask you about it, whether or not you remember or forget it, is more likely to be remembered. So better memory related to a decreased neural response for the old thing. Is everyone on board? Great. And so the specifics I won't get into, but the basic idea is we looked in regions of the brain that responded selectively for this old thing. And we looked at their activity. And on a trial-by-trial basis, we related the amount of activity responding to this to the likelihood, using logistic regression, the likelihood they would remember this competing new item. Okay? And so just to kind of get a little schematic, it's a logistic, so this is a binary. And so a standard sort of effect in the literature is, more activity for something that you process is related to better memory for it. So that's surprising. Critically here, we're predicting the opposite. The less activity there is for this repeated thing, like here, the more likely you are to remember the competitor. So we're predicting negative relationship, negative betas. All right? So from this logistic, we get beta values for every single participant. And then I'm just going to be showing you the mean across participants and comparing them between our experimental condition or control condition. Lo and behold, the reason I'm telling you about this is we found the expected effect in the repeat condition. And critically, this isn't something just about global processing. We don't see any effect in the novel condition, and the difference is significant. Suggesting, again, is something about this asymmetry in mnemonic status, and is something about a decrease in the signal to the old thing that promotes encoding of the new thing. So this is nice and it's good to see, but as with many things in psychology, we want to make sure that's not specific just to, say, spatial attention. We wanted to make sure that it might generalize to other forms of attention, say when the object categories are different from one another. And I assure you, since we're all non-psychologists, this sort of thing is very important in psychology, even though it might seem trivial from an outside perspective. So again, people did the same task. Let me just give you another demo. So people did the exact same thing when they saw images and had to press a button if anything was upside down. All right, good job. That was good. No false alarms. So structure is exactly the same. The critical kind of difference here is we use different visual categories of faces. The scenes were still novel. We're still new, and we tested their memory for the scenes and just varied whether or not this face had been repeated. This is a new face from these two. And so, again, the hypothesis is the decreased activity for faces should lead to better promoting of the scene, as measured by the memory, the subsequent memory. And so, I'm not sure why that's blanked out, but basically this is the repeat face, and that's the novel face condition. That is what we see. So we see this negative relationship here. This is a numerically above chance, but statistically at chance, and a less lower beta for the repeat versus the novel. I don't know where those words went. The other nice thing about this design is because activity in the brain related to scene processing and face processing is spatially segregated, we can also interrogate regions that care about scene information to see what they're doing. And here, our prediction would be because there's decreased activity for this face, there might be relatively increased activity for the scene. And so insofar as this has been seen before, you might be more likely to encode this, so there should be a positive relationship between scene activity and memory for scenes, okay? And so now looking in regions that care about scene information as a function of whether or not that face is old or new, okay? And that's exactly what we see. So the idea is when there's that asymmetry in oldness and newness, it spurs processing of that new information significantly, but when there's not that competition, that sort of processing doesn't dictate things. And then lastly, the nice thing about having these two different selective regions is we can actually directly measure the competitive dynamics between them. So instead of looking at each of these piecemeal, we can enter them basically as a difference from one another. And this is just another form of testing this notion of competition between these two things. So we can account for the decrease and account for the raise using multiple regression, and then parcel that out and look specifically at the difference, the delta between those two things, okay? And so if we look at C minus face, we see this competitive effect. Sorry, the y-axis went up a little. We see this effect where basically when there's that asymmetry in memory, there's competitive dynamics. However, there's not that competitive dynamic when both things are new, okay? So taken together, I've shown you evidence that one form of memory, one neural signal called attenuation, is related to the likelihood that you'll encode new information about the world, okay? And more generally, the idea is that even though, at least in psychology, these two things are often studied in isolation, they're actually very interactive and debatably tightly intertwined. Attention is useful for extracting information about the external world to help sort of jog your memory. And at the same time, the degree to which something in the environment resembles your past experiences, it might help influence where and to what you attend to learn more new information about the world. And again, not to be a broken record, but my whole MO is that understanding these two things jointly is more powerful than examining either one in isolation. Okay, great. So are there any questions kind of about the sort of psych side of things that I just went over? Okay, great. And so for the next little bit, I have just a couple slides looking at more on the methods end of fMRI data. And the goal has been to try to isolate different states. So for me, I care about, for example, different attentional states or states between memory and attention. And the goal of this is basically to separate out two different brain states and do it in a manner where you can kind of do it in a data-driven manner where you can kind of look at all the information at once, figure out what's driving the difference in the two different states, and then go back in and try to tease apart exactly what's going on in terms of, say, particular brain regions. And so I'm not going to definitively answer what is a brain state. This is more just to highlight how I'm operationalizing things, okay? And so in the first part of the talk, I kind of told you a very sort of simple version of what a brain state could be said to be. That is, when you're doing something attention-y, you might get one region of the brain. If you're doing something more mnemonically demanding, you might get another area of the brain. This is a fairly sort of simplistic interpretation, and it actually overlooks a lot of the structure that's actually usually present in our data. And so what we've been interested in more recently has been to look at how activity patterns differ. And so again, just to kind of unpack things is, you can imagine just looking at one region like I did in the meta-analysis to see if it's more or less, right, which is like fine and good. But another way of thinking about it is I can take that entire swath of cortex, and instead of just looking at what's really active and what's not, is you look at the entire sort of topography of activation, okay? And so this little pixelated thing here, this is a stand-in for different sort of voxels, which are the spatial unit of measurement in fMRI data. And so I think throughout when you see my little pixelation here, the idea is these are spatial units of measurement for fMRI. And the notion is, using so-called pattern analysis, you can basically take patterns from different tasks and look to what degree they differ. That doesn't matter where's more active or less active in a particular region. It looks at the overall pattern of activity. And people, have used this to basically disentangle what's cognitive or brain state A versus cognitive or brain state B. We've also been interested in looking at network dynamics. So activity patterns are one thing, right? It's great to know what areas of the brain are showing what sort of profile during what. But just as importantly, for both memory and attention, is how different brain areas are talking to each other. And more practically, that just means how their activities are correlated over time. Okay? So just to get, so the goal from this is I'm going to describe a way that we've been basically feeding in data, trying to figure out what aspects of the data are most important for discerning the two sort of states that we care about. Okay? And just to make it more explicit for those of you who are interested, we compared, for two cognitive states we care about, two tasks. For the pattern analysis, I don't know, just maybe very quickly, raise your hand if you know MVPA. Have you heard of this before? Okay, great. So the general idea is these two patterns are different. And what happened about, over a decade, over a decade ago, is this is just data that you can feed into any sort of machine learning algorithm of choice. And your input is just going to be activity patterns. And I can label these activity patterns with task 1 or task 2. And what I can do is, when I label those, feed them into some sort of classifier. So SVM, you can basically feed it to whatever you want. For this, I'm just going to talk about SVM. You label these, right? And you build up your classifier so that it gets really good at discerning how, in what way, two patterns of activity are different from one another. And then you test it on some left out data, and you say, how good of a job did it do? And if it's that chance, you're saying, well, I guess these patterns are not very distributive. However, if it does a really good job on, decoding or classifying that other information, the argument is that basically you're picking up on two different neural patterns, two different states. All right? Everyone good? Great. Here, even though we're kind of taking all our voxels and we're just kind of tossing them into the soup, which voxels end up being more or less important is actually really important for science reasons. So, different regions might contain sort of, might provide more discriminative information for the classifier, and where that falls in the brain is actually super important. Now, for networks, we've kind of taken a different approach. We do a very analogous analysis, and I think, at least in the talks I've seen here, oftentimes people will have these kind of nice noded edges overlaid on some brain, and we're kind of doing one step more ambitious than that, which is, in that sort of information, people often lump a bunch of voxels, and they call it a region, and they make it into like their little node, and then they just draw edges between them. We're interested in basically looking at every single voxel, okay? And the idea is that, in a particular MRI volume, in a particular acquisition, there's about 30,000 or so voxels. So oftentimes in, fMRI network talks, they try to reduce that down. They reduce the number of features. They reduce the number of spatial units down to 1,000 or a couple hundred, or a couple dozen. Because we're collaborating with some folks in the parallel computing division at Intel, the idea is to just use brute force and not compromise and just be very, in some ways, wasteful without not making any assumptions, but using a lot of data and then trying to recover on the back end what's driving things. And so here, when I talk about networks, basically what I'm referring to is a voxel-to-voxel correlation matrix. So here I fed in activity patterns, just the amount each voxel is activated. Here what I'm doing is I'm taking the upper triangle of a 30,000 by 30,000 correlation matrix, right? Whole brain correlation matrix. And I'm going to do this same little trick of classification on that correlation matrix. All right? So again, same drill, labeling them, training, testing on left out data, And the notion is, insofar as you're able to discriminate between two things, that's suggestive that you're capturing some difference in the brain states. And so and also here, which features? So here I'm going to call them edges. These are just entries in that correlation matrix. I don't know which language is safe to use around here. I just think of these as basically entries in the correlation matrix. They're Pearson R values between correlated activity in different parts of the brain. Figure out what edges, what entries in the correlation matrix are kind of doing the interesting things. All right? And then the goal is to do it for both of these things, see how well it does, and also explore ways in which they might interact. Okay? So it's very early days. So in particular, the FCMA approach has been done in terms of published work on one data set. We've been playing around with another fairly basic data set. The task is you just have blocks where participants lay in the scanner, they look at this little plus sign, and they do some tasks either on something to the left of that plus sign or something to the right of that plus sign. Like super boring, but great, because the idea is you can try to train up a classifier on distinguishing basically leftward versus rightward attention. And we know from many, at this point, decades in neuroscience, that there are meaningful neural differences between these two things. So it's a good sort of proof of concept space. And so far, so good. So what we do is, I can talk more about details, but just using kind of broadly construed these two different approaches, we get very good off-chance decoding accuracy. So the idea here, this is just kind of, it's not maybe not familiar with various terms, but this is kind of the cross-validation accuracy decoding leftward versus rightward attention. And so if I use MVPA, that is, I take the pattern of evoked activity, I get like 85% correct, which we're happy with. What's kind of cool is we also get well off chance, these are not significantly different, decoding for FCMA, meaning that there's a lot of information to be had in the correlational information as well. And as I alluded to before, one thing we're really interested in is this all the same underlying things? Is this correlation driven by the changes in activity? We can't get directed graphs, we can't tease things apart, we're just looking at things that are correlated with each other. And so what we've been doing is basically combining the predictions from classifiers. So you get a guess from your MVPA classifier, you get a guess from your FCMA classifier, and there's different ways of kind of bagging those together, of kind of combining those to make a single guess. And what's neat is, it's not visually striking, but I assure you this is a significant improvement, is that if you combine the predictions from both forms of classification, you do slightly better. And this is, again, early days, but suggestive that there's benefit to be had from considering both sort of the evoked response as well as the structure in that correlation matrix. That is, they can add things above and beyond the contribution one.
So, could you repeat how the MVPA differs from the
Sorry,
and then you said that the FCMA is using the voxel to voxel thing, but what was the MVPA?
Yeah, so the MVPA is basically the pattern of, sorry, not anything more descriptive, but this is actually kind of useful. So it's basically the pattern of evoked response. And so during a block of leftward attention, the data I'm showing here is from about, we did some feature selection, which is almost like a different talk, but like, the idea is we're looking for MVPA for about 2,000 voxels. And we found those doing kind of a nested cross-validation of this approach. But within those 2,000 voxels, for each sort of block of leftward attention, you get a vector, 2,000 length vector, different intensity values, and you have those for both leftward and rightward attention, and you disentangle them and apply it on left output.
So the only difference between the FCMA and the MVPA is that in the MVPA you're actually first doing the feature selection before you do the correlation?
So both of these you do feature selection first. The key difference is just the contents of what those features are. One case is the evoked activity, or the bold measure within that voxel, and then the other case it's you can picture the 30,000 by 30,000 correlation matrix. There's entries in that, which is, for now, the only distance metric we're using is just Pearson's R. And we're just very kitchen sink approach. We're tossing it all in, and we're using, we're reducing that correlation matrix down to a vector. And then you have a vector for all your blocks of leftward, all your blocks of rightward, and you do classification on that. And for the FCMA, that's after feature selection, it's about 1,000 by 1,000 voxel correlation matrix. And great, so actually that brings up one of the things. So this is all fine and good, but as I alluded to before, we're really interested in where in the brain is driving this. You know, it's nice to know you can do it. That's obviously the first step, but it's less useful from a basic science question if you want to understand what is sort of the underlying structure of these different brain states. And so ongoing work that I've been mostly interested in has been trying to tease apart what's going on in the FCMA results. So I know, for example, I have a good sense of what voxels are important for discriminating between two things. This is just, I call this a spaghetti plot, but the general idea is I can take the more informative voxels and I can look at kind of the most, the correlation values with kind of the highest difference between the two conditions. And I can generate something that kind of looks like this, and it's mildly interesting, but it's not quite capturing what we think that sort of classification is picking up on. And so one thing that I'm interested in is getting a better sense of going from that correlation matrix to understanding what aspects of it are really driving performance, how they kind of spatially cohere to each other, in what ways are they meaningful units, and that in turn will kind of inform what sort of pathways or what sort of pairs of brain regions are actually doing the heavy lifting. Also, as you might have picked up on, we're currently just using a correlation matrix. We're very much interested in kind of expanding the metrics we use, whether kind of branching out to different distance metrics or trying to take that correlation matrix and either recharacterize it or reduce it down in different terms, in terms of aspects besides just correlation, to see if there's any other ways of describing that network that are interesting and equally powerful at discriminating the two states. All right, great. So I think that's all I have, and I'm happy to talk more about anything. Just this is the work in the first half was for my grad work. The work in the second half was when I did my postdoc, and then the last little bit is collaboration with folks in the Turk Brown Lab and Intel Labs. Thanks. Any questions? You don't even have to be psychology related.
So a lot of the stimuli that you're using, especially like the visual map stuff where you can get sort of on the cortex and draw the different polar angles. Is there anything about visual information or that is special about that, or can kind of any sort of information content?
So the maps that I showed you in that region of the brain, truth be told, don't actually even rely on visual stimulation. So they're thought to really, I didn't stress this, but they're thought to actually track top-down attention. The idea is, say I give you, in the middle of the screen, I say, in about 10 seconds, look to this location in the periphery. It turns out that you attend to places before you look to them, and you can get these maps just based on that. Even though there's no visual stimulation, the plan of where you're going to saccade to has systematicity in terms of that spatial information. And so for those maps, the visual content actually doesn't matter. In fact, it doesn't even presuppose visual stimulation. But there's many other regions of the brain where the opposite is true.
So sorry, you say that you're actually not looking at the regions which are supposed to track down specific objects in the scene. You're more just looking at the regions that are about attention.
Yeah, in the part with the visual maps along the IPS. Yeah, that's right. But they're very related to each other. And you know, I gave an example of how you can really tease apart just the sort of top-down signal in the absence of bottom-up stimulation. But more meaningfully, you know, those things actually integrate both sort of top-down signals and bottom-up information in order to guide behavior.
You were saying something about you know, your choice of faces versus scenes.
Yeah.
Because you know, the face recognition usually is different from what is recognizing scenes.
Yeah.
But do you also, how do you make sure that the features in the scenes, you're showing, for example, two different pictures of scenes, but you don't care about anything that will be about because of similarity in the scenes? You're not worried?
So this, no. so this I'm sweeping in under the rug, but those sorts of things I'm very interested in. And so, for example, again, I don't know how familiar people are. This is often called in the literature fusiform face area, or FFA. It's a little bit of a loaded term because it cares about other things as well. This red splotch I'm showing you here is sometimes called PPA, parahippocampal place area. And the idea is if you just look at evoked responses, if I show you a scene, these regions are going nuts and these regions care less. If I show you faces, these regions are going nuts, these regions care less. That's the principal response I'm kind of harvesting for this study. At the same time, there is structured information in that response. So using these pattern analysis approaches I used in the kind of new bits, there is similarity structure actually in these regions of the brain. So if I show you, if I feed this whole area and look at the pattern of activity, treat that as like a vector, and I show you a picture of a spatula and a picture of like a hairbrush, you get fairly similar patterns of activity. If I show you a picture of a spatula and a picture of the opposite of the spatula, like a pancake comes to mind, but a semantic. Anyway, some like large rotund structure. The idea is you get very different patterns, and you can look at that similarity structure. There's really cool work by Nico Kriegenscourt and others where that structure of representational similarity in visual cortex is very similar between species. So for example, monkeys have very similar visual representational structure as humans. And kind of moving forward, when I'm really interested in the internal attention bit that we didn't find evidence for. But one aspect of understanding what internal attention is understanding the space in which it operates, which is understanding that similarity structure. Yeah, sure.
So when you have the inverted U shape with the different categories of like depth of remembering, I guess attention, with reaction time, do you have a sense of how? And you were saying that it kind of corresponds to uncertainty in like memory retrieval or some sort of fuzzy memory. Do you have a sense of how that would look in a network context?
You mean a network of like brain region to brain region?
Yeah,
sort of
really
activity?
vaguely. Like is it sort of a lot of like imprecision in between?
Oh, I see.
In between regions.
So it's a very good question. The short answer is I don't know. You know, there's work having to do with perceptual decision making more generally, where it makes predictions, for example, about the strength of the amount of information transmitted between region A and region B. But I know less about how that precision kind of flows up through the system, which I think is more about it. There has been some work related to this in non-human primates by Bill Newsom and others, where you kind of can get neurons that care a lot about the choice, and by doing similar, almost sort of pattern analysis type things on those neurons, you can get information about how there are different patterns for kind of different levels of confidence, which is kind of related. Yeah.
Okay, no more questions.
All right, thanks.